Welcome to The Explainer. Today, we're diving into something that's absolutely everywhere in computational biology right now, AI virtual cells. But look, we're skipping the sensational headlines. We want to look at how this tech actually works, and more importantly, how we cross that massive gap between just predicting what a cell does and actually controlling it safely to cure real diseases.
Okay, let's jump right in. We really need to ground ourselves in biological reality here. I know it's super tempting to look at AI and expect some instant miracle cure, but as this quote nails it, disease remains a system-level failure, not a single switch problem. We're not dealing with simple computer code where you just patch a bug with a quick fix. Biology is messy, it's incredibly interconnected.
So, to unpack all of this, here's our roadmap for today's explainer. First, we're looking at why disease is a system collapse. Then, we'll cover the virtual cell revolution, why prediction is not control, the EP control framework, and finally, how we actually make medicine governable. Don't worry if this sounds dense, we're going to walk through all of it together step by step.
All right, Section 1. Disease is a system collapse, beyond the single pathogen. We really have to understand that complex diseases are not simple infections with one clear enemy.
Think about things like Alzheimer's, cancer, cardiovascular disease, autoimmune disorders, stroke, or metabolic degeneration. These aren't simple. They emerge from incredibly dynamic networks. We're talking interacting webs of genes, immune signals, metabolism, tissue states, inflammation, aging, the whole shebang. So what does this actually mean in practice? It means two patients with the exact same diagnosis might have totally different underlying mechanisms driving their illness. For one patient, it could be inflammatory dysregulation. For another, metabolic stress. And that variability makes relying on the old drug discovery paths pretty unstable. And that brings us right to why the traditional drug discovery model often fails. It's usually built on a super narrow hypothesis, right? One target, one molecule, one trial. It goes after surface symptoms, like the decades we spent just focusing on clearing amyloid and Alzheimer's. Sure, it produced knowledge, but not a clean, universal cure. Why? Because it completely misses the system-level failure. It assumes biological states are uniform across patients when really they differ wildly across subgroups. You literally cannot fix an entire network by just flipping one switch. Which brings us to Section 2, the virtual cell revolution, and the sheer scale of ambition here. This is where AI steps in and brings a totally new toolkit to the table to tackle this complexity. This whole movement rests on three massive technological pillars. Number one, measurement. Things like single-cell RNA sequencing let scientists observe exact gene expression states right down to the individual cell. It tells us what a cell is doing right now. Number two, intervention. Using tools like CRISPR, researchers can systematically change genes, moving from just passively watching to running causal experiments. And three, prediction. AI virtual cells take all that massive biological data and learn to predict exactly how a cell is going to respond to a drug, a signal, or a genetic change. Now, taking a look at this, the core thesis is absolutely vital to grasp. The decisive problem in AI biology isn't just whether AI can predict cell states. The real problem is whether that prediction can become reliable, causal, safe, explainable, and patient-specific control. Just predicting what a cell might do, that's not medicine. We have to be able to govern that change. To actually get closer to that goal, the sheer scale of ambition right now is just staggering. 1 billion. That is the number of high-quality cell experiments the ARC Institute's Virtual Cell Initiative is aiming to conduct. Basically, they want to scale up causal biological data to a level we've never seen before, dragging biology out of isolated lab experiments and into industrial-scale data generation. And they are making massive strides. The ARC Institute released a first-generation model called STATE. The scale of this model is wild. It was trained on nearly 170 million observational cells and over 100 million perturbational cells, spanning 70 different cell lines. But hey, the real test isn't just memorizing lab data, right? ARC also launched the Virtual Cell Challenge to test generalization. Can these AI models accurately predict responses in totally new, unseen biological contexts? Because that is the true test of a virtual cell's utility. Moving on to Section 3. Prediction is not control, the crucial gap. This is where we have to separate science fiction from clinical reality. Look, there is a lot of exaggeration out there. The hype will tell you humanity is going to have no disease in 4 years, treating human illness like it's just a software bug waiting for a patch. But the reality? Yes, virtual cells are incredible for accelerating target discovery, disease subtype analysis, and early drug screening. But they are not instant clinical cures. Real patients have entire organs, immune systems, microbiomes, and complex histories. An AI predicting a single-cell change does not automatically equal a safe clinical treatment for a human being.
So here's the absolute crucial point. Prediction just estimates outcomes. It tells you what might happen in a vacuum. Control, on the other hand, determines safe, effective action under strict constraints. Control is deciding what to actually do with that prediction when a human life, with all of its messy complexity, is on the line.
And biology makes control incredibly difficult because, frankly, biological systems are stubborn. They adapt, they compensate, and they reroute. You might use a drug to fix one signal, but it completely destabilizes another. Tumors adapt. Immune responses can wildly overshoot. So our primary job isn't just picking a gene to push, it's figuring out how to carefully guide a complex system toward health while actively minimizing any risk of secondary damage.
So how do we actually do that? That brings us to Section 4, the EP control framework, bridging the gap. This is the essential missing structure that turns black box AI predictions into safe, actionable medicine. EP stands for the energetic paradigm. Basically, it's a reasoning framework that maps disease as a dynamic energy and control problem to safely guide intervention. It isn't just some basic safety checklist tacked onto the end of an AI model, it's a comprehensive architecture. It maps out causal structures, boundaries, and system behaviors. It looks at the biological system and asks, okay, where are the flows, the bottlenecks, the loss channels, and the governing constraints? To really get EP, let's look at its core rules. First, disease state mapping. It finds the true causal drivers and recovery paths, not just surface symptoms. Second, causal diagnosis, which forces us to separate mere correlation markers from actual levers we can pull. Then it creates an intervention grammar, giving structure to the sequence and timing of actions. It also builds an evidence architecture to handle uncertainty, applies patient-specific reasoning, and provides the translational discipline needed for clinical trials. In short, it's the logical grammar that turns mysterious AI outputs into accountable clinical actions.
In practice, an EP-controlled workflow is basically a multi-layered filtering pipeline. You start with pathology and constraint mapping, then you move to causal decomposition. Only then do you use virtual cells to search for interventions. And crucially, this is followed by EP pruning. This is where you actively reject any interventions that violate safety limits or causal logic before they ever get near a patient. Finally, everything goes through an adaptive validation loop.
All right, finally, Section 5, Making Medicine Governable, the Future Landscape. Let's look at what this all means moving forward for clinical medicine.
Now, what's really interesting here is comparing the two approaches. Using an AI virtual cell alone just gives you black box confidence, weak generalization, and a really hard time translating to the clinic. Honestly, it's basically a highly advanced guessing game at scale. But an EP-controlled virtual cell, that provides explicit uncertainty tracking, causal audits, constraint discipline, and adaptive feedback. It converts a raw prediction into causal constrained intervention logic. It makes the whole discovery process governable and explainable.
And this leads to a completely new medical ecosystem. Big Pharma isn't going anywhere, but we are going to see a new division of labor emerge. AI biology platforms are going to handle the design and discovery of interventions. The energetic paradigm will sit right in the middle, providing that vital control, logic, and integration layer. And pharma will take those safe, rigorously pruned candidates and scale them up through clinical execution and regulatory pipelines.
Which leaves us with this final thought to chew on. AI is giving us a powerful new simulator for human biology, a high-resolution window into the cell like we have never, ever had before. But a simulator is just a tool. Are we ready to use the energetic paradigm as the disciplined navigator we need to actually safely cure disease? Because without it, virtual cells remain just fascinating predictors. But with it, they become part of a governed system capable of truly transforming human health. Thanks so much for joining me for this explainer. I really hope this helped you grasp the incredible reality behind AI virtual cells. Keep learning, and I'll catch you next time.